Hello, my name is Professor Kwaku Ohene Frempong. I'm the program coordinator for the National Newborn Screening Program for Sickle Cell Disease in Ghana. By training, I'm a pediatric hematologist, and for years I was at Children's Hospital of Philadelphia, from where I began to develop the newborn screening program in Ghana. As you've heard, newborn screening starts with pregnant women who need some education about the whole process of newborn screening and how it's important that their children be tested. And for those that we find to have sickle cell disease, how it is important that they enroll in a clinical program that will protect those children with early intervention. The laboratory aspects of newborn screening are relatively simple. We require that we receive samples from all the babies that are supposed to be screened and quickly process the blood samples to be able to get results that are meaningful and to return the results to the sites where the blood samples were obtained or other sites where reporting will take place. And we know it's important that as quickly as possible, the families need to learn whether they have a baby with sickle cell disease, who then need to be enrolled in a clinic very quickly and started on treatment with penicillin prophylaxis. Those who don't have sickle cell disease also need to know what the hemoglobin types of their babies are. The urgency is not as great because there is no clinical intervention that is needed. When the samples are received in the laboratory, nurses have already obtained information about the baby, about the family, where the family lives, and contact information such as telephone numbers for the family members. The task of the lab is to first ensure that the information about the baby has been fully collected, that there is clear identification of the baby, the family, where the family is located, where the sample was collected and at what time it was collected, when it was shipped, and then record for themselves when the sample was received. The laboratory procedure that we use in our lab at Noguchi Memorial Institute for Medical Research in Ghana is isoelectric focusing, or IEF. It's a very simple method that enables us to determine whether the baby has any abnormal hemoglobins, especially hemoglobins S and C, which are the primary targets of newborn screening for sickle cell disease in West Africa in order to diagnose the babies with the disease. The test itself is relatively simple to do. It takes about 2 hours from the moment the samples are being prepared to the run and when the results are read. The lab can sample about 66 babies' blood for each analysis. So in a day, if they have 3 runs, they are screening about 200 babies or testing about 200 babies. And the lab is actually equipped to do even more samples than that. They use a dual system of ensuring that the results have been checked. So one laboratory technician who did the runs will record what he or she thinks the results show, and a second lab technician looks at the same work and checks over the writing, the results that were written by the performing technician to verify that the findings and the recordings match. And once their work is completed, the sheets on which they recorded the results then get sent to the data entry clerks, who by this time are also entering information about the baby, and then that information is matched with the results. It takes about 2 or 3 days for them to complete all of that work. And then after they've checked and verified that their work has been done properly and the recordings have been made properly, they send the results to the screening sites. For every baby who is found to have sickle cell disease, the lab will actually take another sample from the original filter paper and rerun the test to ensure that the results that have so much implications are actually correct. So they do verify the first sample before they send it. Ideally, when the baby then gets enrolled in clinic, a second sample is supposed to be obtained and sent back to the lab for the lab to reconfirm, in this case, to establish that the identity of that baby whose first sample was found to have sickle cell disease is the same as the child who has come back now to start on clinical management, because we don't want to have an error in the initial sampling where is the wrong baby sample that was identified with the name of that baby to ensure that we don't put the child who doesn't have sickle cell disease on treatment. So the laboratory aspects of it are relatively simple. I think the harder work is how that information then begins the clinical activity to protect the child with sickle cell disease.